Now we are going to talk about protein structure. The structures of proteins are actually extremely large compared to simple organic compounds. To better understand protein function, we must look at the four levels of organization for their structure. These are defined in the following way. In proteins, the organization level for structure is divided into primary structure, secondary structure, tertiary structure, and quaternary structure. Let's look at each of those levels individually. Starting with primary structure of a protein, understand that primary structure of a protein is basically the linear sequence of amino acid residues that are found in a protein chain. We learned previously how to draw protein chains, and we observed overall that whenever we had to draw a peptide, specifically because in this class we're not going to be drawing proteins because we are not going to be drawing 51 or more amino acids altogether. What we have is the presence of an amino side, the alpha carbon with its alpha hydrogen, and then an R group coming out of it. Then we have an amide bond, and then that sequence repeats up until at the end of our peptide, we have the last alpha carbon with its side chain, and this area tends to be a C double bond O, O minus overall. The area where we have the pattern NH-CH amide bond, CH amide bond, CH amide bond, all the way down to CH, C double bond O, O minus. That is known as the backbone of the protein. And you can see that it is written in color. We can actually draw a 3D version of that. And that means that what we are incorporating is the stereochemistry that is present in the alpha carbon, specifically in peptides. So if we start with an NH group, and remember that it could be written as NH2 if you start with proline, or you can write it as NH3 if you have any other amino acid. Immediately we have the alpha carbon. And if that vertex is pointing up, then we tend to write the side chain as a wedge. If, as you write the rest of the molecule, it happens to be that after the amide, we land on the vertex that specifically is pointing down, we tend to write that side chain as a dashed line. And overall, as we move along and we draw this structure incorporating the amino acids, then understand that that's where the wedges or dashes are represented. This is more to illustrate to you that at times you will be provided peptides in this form. Going back to the primary structure of a protein, we can observe as an example in the right side of the slide, this is a small segment of the amino acid sequence that it is present in the B chain of insulin. As I previously mentioned, insulin is an important protein that is found in humans and other organisms. Overall, the structure of insulin is composed of two chains linked together by disulfide bonds. We can see in this segment that we have the drawing of the sequence specifically between amino acid 15 and 23. One of the things that it is important that you know how to do is that if a peptide or a primary sequence of a peptide is given to you, you can define what are the amino acids present in it. Let's practice on how to do such exercises or problems. So you can observe here that we have two versions of the same. exercise. Some can be found in homework, while others can be found in exams. So, looking through the first example, I'm going to zoom in so we can look at this closer together. In this primary sequence for this peptide, you can see that overall, how we have this written, this is the N terminus of your peptide, because overall, this area represents the first amino acid. And all the way to the left of my peptide, I have the C terminus. And in the C terminus, understand that I have another amino acid. Following the pattern that we learned when we were going from amino acid sequence to drawing a peptide, now basically we're reversing that process. We're going to find the pattern that allows us to find the alpha carbons in our peptide. So when we look closely here, we can define that we have specifically 1, 2 peptide bonds. That means that specifically in this problem given, I have 1 amino acid, 2 amino acids, 3 amino acids. Notice that I'm just highlighting the alpha carbon because at the alpha carbon is where I have each of my side chains so I can identify the amino acids present in the peptide. Starting with the first one, which is a CH connected to a CH3 and a CH3. This side chain, if I actually change it into skeletal form, basically means that I have the side chain that looks like this. And I'm just going to put a squiggly line. Above the squiggly line basically is where we have this CH. So if you look through the table that is given to you as a resource, you can notice that specifically that corresponds to the amino acid that is called valine. So make sure that you read the instructions carefully when you are taking exams to see if you need to write the 3-letter code or the 1-letter code. For now, I'm going to write both the 3-letter code and the 1-letter code. So for valine is VAL, or in other words, V. Next, I'm going to look at what is the amino acid in which its side chain is a CH2SH. As I go through my table, I see that that amino acid actually is cysteine, which is 3-letter code is CYS. And the single letter code for it is specifically the letter C. Next, I have a side chain that looks specifically like a CH connected to a CH3 connected to an OH. Let me write it in skeletal form. Remember, the squiggly line basically is this area. And coming out of that CH group, I have a carbon connected to CH3 connected to OH. When you look at your reference table, that technically is the amino acid that we know as threonine. Its 3-letter code is THR, and its 1-letter code is the letter T. Let's look at another example of how to determine the amino acids present. So I'm just going to overall format the answer. So I'm going to write val cis THR V connected to C connected to T. That's how you format your final answer. Let's do the next example in which we have then a different way to write a peptide. So when it comes to this, you can highlight if that helps you, the amide bonds. I am just going to put dots to actually separate where the alpha carbons are. So this is my first alpha carbon. Then I have an amide group. Then I have my next alpha carbon. Then I have an amide group. Then I have another alpha carbon. Then I have an amide group. Then I have another alpha carbon, amide group, alpha carbon, amide group, alpha carbon. And we can observe that this is where we end with C double bond O minus. So that's where our peptide ends. So now at those blue dots, that's where we have the side chain. So that means that specifically this peptide is going to have 1, 2, 3, 4, 5, 6 amino acid residues. So starting with this side chain, that actually corresponds as you look through your table to tryptophan. Let me do the 3-letter code. That will be easier. So 3-letter code is TRP. 1-letter code is W. Next amino acid. Technically, these dashed lines represent just having a CH3 at the end. So this represents ALA. 1-letter code is a capital A. Both of these side chains are the same. It is a CH2 connected to benzene. CH2 connected to benzene. When I look through the reference sheet, this actually refers to phenylalanine. So at this site, I have PHE, 1-letter code is F. Here is another PHE, 1-letter code is F. Next amino acid right here is a CH2 connected to a CH and two CH3s. That is isobutyl. Isobutyl specifically is leucine. So the 3-letter code is LEU. Capital letter is L. And then lastly, I have a CH2, CH2, and a carboxylate. When I look at my table, that actually corresponds to glutamic acid, in other words, glutamate. So the 3-letter code for, let me go back, this site is actually GLU, and the 1-letter code is capital E. So when I write my final answer, I write TRP, ALA, PHE, PHE, LEU, GLU, and that corresponds to W, A, F, F, L, E. You may have noticed that that second peptide actually spells a word. In your homework in WebAssign, you're not going to have peptides that spell words, but please expect that in examinations, in a way to make it fun, I will be presenting specific peptides that will be overall spelling words. Let's now move on to look at the next organization level for protein structure, which is known as the secondary protein structure. When it comes to the secondary protein structure, the arrangement of protein chains into these patterns come from hydrogen bonds that are going to happen between amide groups and carbonyl groups of amino acid residues in the chain. So that means that the secondary structure for proteins actually comes from hydrogen bonding events that happen in the backbone of your protein. The structures that result from these hydrogen bonding events are known as alpha helices or beta pleated sheets. In an alpha helix, which is what we have specifically in figure 12.10, what we see is, in essence, hydrogen bonding events that form like a helical structure. So you can think about it like a winding staircase in which basically this ribbon that is going around, that's basically the backbone of the protein. In the case of a beta pleated sheet, this secondary protein structure are going to happen because you have protein chains that are aligned side by side in a sheet-like array. And again, it doesn't matter if it's alpha helices or beta pleated sheets, hydrogen bonding events that are happening between the NH group in the amide and a neighboring, usually 3.5 to 4 residues down, specifically for the alpha helices. And then we have other arrangements for the beta pleated sheets, is what results in specifically the formations of these protein structures. Next, we have the tertiary structure of a protein. The tertiary structure of a protein is specifically the three-dimensional shape that a protein adopts as a result from the interactions of the side chains of the amino acids residues in the protein. There are different interactions that are present in the tertiary structure. They include disulfide bridges. Disulfide bridges only happen when two cysteine residues undergo oxidation and they form a disulfide bond. Then we can have salt bridges. Salt bridges is an ionic interaction that occurs between amino acid residues. Here we can see that if you have an ionized acidic amino acid paired with an ionized basic amino acid, this ionic interaction is what we call the salt bridge. We can have hydrogen bonding events happen between the side chains of amino acids. So you can imagine if you have amino acids that by definition have a hydrogen connected to at least oxygen or nitrogen, and it is in the presence of another side chain that has oxygen or nitrogen, then hydrogen bonding events can result. So you can see here in the example that serine interacting with serine, since both side chains we have a hydrogen connected to an electronegative oxygen, and that is true of both, we can see in this dotted line the illustration of a hydrogen bond. Lastly, we can have hydrophobic interactions that can occur in the tertiary structure of a protein. When it comes to hydrophobic interactions, they result when nonpolar groups are either attracted to one another or forced together by their mutual repulsion to an aqueous solvent. So what that means is that typically a hydrophobic interaction, which basically by definition means that these are going to be non-polar side chains in amino acids, and we can see the interaction of phenylalanine and phenylalanine interacting. Since these side chains are non-polar, they can interact with each other due to dispersion forces. And since the polarity of these side chains is opposite from water. Remember that water is polar. Then that's what create a hydrophobic interaction. Hydrophobic interactions, interestingly, tend to happen and be stored in the interior of proteins because those residues, when the protein is folding, it actually wants to be away from water as possible. So that's why hydrophobic interactions tends to be in a protein in the interior of it. We can now observe examples of prediction for if we have our groups interacting, can we define actually what is the interaction that they will have if they were in a tertiary structure? So, for example, if we look at the first pair, which is the side chain that it is an isopropyl group versus the side chain that is a CH2 benzene. In essence, we are talking about what will happen if the side chain of valine is interacting with the side chain of phenylalanine? Since both of these side chains belong to the nonpolar side chain group, that means that their interaction in the tertiary structure is going to be hydrophobic. Next, we have specifically a CH2CH2COO versus a CH2 with this heterocyclic amine. When we look closely at who do those side chains belong to, the first one belongs to glutamate, in other words, glutamic acid, which its three-letter code is GLU. And then the second amino acid actually belongs to histidine, which its three-letter code is HIS. If we look closely at what is the classification of these amino acids, this is an ionized acidic amino acid. This is an ionized basic amino acids. So their interaction in a tertiary structure would be via a salt bridge. Lastly, we have the side chain of an amino acid that is CH2OH. We have the side chain of an amino acid that is CH2, C double bond O, NH2. When we assign which amino acids have these side chains, the first one is serine. The second amino acid is going to be asparagine, meaning asin. So, what would be the interaction between a serine residue and an asparagine residue? Well, in the case of the serine, we actually have a hydrogen connected to oxygen. In the case of the asparagine, we have the electronegative oxygen. So these two amino acids can interact via hydrogen bonds, in other words, hydrogen bonding. To conclude looking at the organization of protein structure, the last thing that we have is the quaternary structure of proteins. All proteins have a primary structure, a secondary structure, and a tertiary structure. Quaternary structures are actually special. Quaternary structures are the arrangement of multiple tertiary structures that are also known as a subunit, and that forms a larger protein. Basically, in a quaternary structure, subunits are going to be held together by various attractive forces that can include ionic attractions, disulfide bridges, hydrogen bonds, or even hydrophobic effects. So, when it comes to a protein that has a quaternary structure, understand that even though it is made by subunits, each subunit is going to have its own primary structure, secondary structure, and tertiary structure. An important structure in human that has different subunits is going to be sorbitol dehydrogenase. Sorbitol dehydrogenase is a protein that is responsible for converting sorbitol to fructose. And we can see its crystal structure, which this is a computer generated structure for this protein. And in figure 12.16, we can see the four different subunits that make up this protein. You can see them in their color coded. So here there's one subunit, here is another one, here is another one, and this is the last one. So sorbitol dehydrogenase is an example of a molecule that has a quaternary structure.